we'll record right here. Okay, so the recording started and we can kind of get going a little bit and talk about some things. Again, as I said, I hope all you guys are doing okay. It is a really brutal time, I think, for everybody. The burden of learning.
I got it.
There? Good job. I think the burden of learning has always been with you guys, with the student at the university. And I think all of the focus on distance education and these Zoom meetings have really emphasized the degree to which the burden is really on you. And it's not sufficient just to show up and have me read the textbook to you. And certainly when I was in college, that was kind of the routine. You'd go to class and somebody'd stand up at a podium with the textbook in front of them. And they would basically read from the textbook. Periodically, good professors would use a blackboard, but there was no such thing as Microsoft PowerPoint, or really the only place we ever saw slides was when I took histology. And basically at that point in time, we had an old Navy captain taught my histology course, and he would stand at the podium with a clicker and he would click the slides, and he would sit there and, slides of cells and tissues would come in back, and he'd stand there. And of course, I'll use myself as an example. He used to call us by name and he'd say, "Barbato, what's this tissue?" And you had to stand up next to your chair and then say, "Oh, it's simple squamous epithelium, Dr. Rife." And he'd say, "Correct." And he'd click again. Of course, I got a few wrong here and there too, but that's okay. And now we're in but it was really up to you. It's always been up to the student. And I think, unfortunately, for a lot of students, not all of you, I think most of you guys who are here are, real solid citizens. But I think college itself is a real shocker, the degree to which it's really not up to the teacher to actually teach. It's up to the student to learn. If you think about the origins of the university, basically all we did was give exams. And we said, "Okay, you're taking genetics, here you go. When you're ready to take the exam, let me know." And then you took the exam, and the exam certified that you knew enough about genetics that the faculty decided you needed to know to pass. So there were no lectures in the original European model, which is what all American universities are based on. It's actually German model. But this switch has really changed everything and really put the burden on you guys. And I think the stress of it shows on all of us. I think the stress of it shows on all of us. So this is all kind of my lead in to say, I am trying to be as flexible as possible in the way we approach of learning the assignments. Nobody will ever get a late, I'm lowering your grade to a B because you were late. Nobody is late. There is no such thing as late unless you wait till December 14th to submit everything, okay? I would prefer that you kept up with the blog postings, only because, in fact, that may be the only time that I might ding you some points, is because I really don't want to read 36 times 7 is blog posts. I prefer not to read 250 some odd blog posts, just before Christmas. Not that your writing is so depressing, but it's just too much. We should be kind of keeping up to the best of our ability. But as I said, nobody is going to get dinged for late assignments. All right. We are going to try to work through these the best that we possibly can. I appreciate all the feedback from everybody. And we're going to continue these Friday Zoom sessions. every Friday except for the Friday after Thanksgiving. If someone really wants to do the Friday after Thanksgiving, I will have a computer, but you do need to bring me a turkey sandwich if I'm going to do that. That's really important to me on the Friday after Thanksgiving. So we need to have turkey sandwiches. So just to kind of keep up with that, we'll be doing this. I will make sure and record them and make sure that the recordings are available for you if you cannot make it on a particular Friday. Okay, these are not mandatory, they're kind of for us. So I want to go over a couple of things. It's going to take me a moment to kind of just do a few things that I think will be helpful. And if I can get to my Zoom.
Professor Barbato, I have a
Yeah.
question. Do we have an exam coming up in like a couple weeks?
Probably.
Okay, just wondering for sure or not.
Probably. No, we're going to try to stick to our, I know that Dr. Luke is planning on an exam October 3rd. I think I'm looking more toward the following week. So figure, like at least 2 weeks. I want to make sure that we've all had an opportunity to get through these couple of problem sets. And other than the chapters 1, 2, and 3, which should be completed by now, we're getting ready to move into a couple of the other chapters, non-Mendelian forms of inheritance. And we'll talk a little bit, but the problem sets I think that we're going to talk about today are, I think, the more relevant and will comprise about half of the exam. But I want to make sure that we have enough time that everybody is on board. Okay? Make sense?
Do you know like which chapters are going to be on the first exam other than 1 through 3?
1 through 3, and we're now approaching into 5, which is I believe is non-Mendelian forms of inheritance. And we'll be talking a little bit about pleiotropy and epistasis. So, all the topics that we'll be talking about from here. I am going to avoid anything having to do with the molecular biology of the gene, or what I prefer to call the biochemistry of the gene, until the next section. So the exam will basically cover everything up unto the biochemistry of the gene.
So we're not following the chapters on that list of...
I think it's 5, 8, 11. I don't recall. I don't have it in front of me offhand.
Yeah, it was 1, 3, 2, 8, 11,
1, 3, 2, 5, 8. Does this sound familiar? Well, actually,
yeah.
This
I just want to make sure.
Yeah, this leads me to a topic that we'll talk about here right now. You should see my piazza on the screen. And one of the things is, of course, by now, there are actually a few people who have not signed up. They've not joined Piazza. I don't know if you're planning on dropping, if it's you. I doubt anybody who's here is actually one of those people, but I really haven't looked at a list right now. But please, this is where all of our communications really come from, except for the periodic Blackboard email. Up top, if you look at the top row here, of course, you've probably all found the Resources tab. And the Resource tab, right under Bio 210, has this thing called the syllabus. And if you read the syllabus, hopefully, did everybody see the Stockton syllabus here? Or no?
Yeah.
Okay. So, sorry, this is on my Mac computer. And if you kind of poke down, there's the occasional comic, all the crazy stuff. Yeah, so here you go. 2, 8, 11, 3 to 5, which is the basic principles of inheritance, recouping 3, and then, of course, pedigree. So up to 6. And we will probably talk about a little bit about sex determination. And then that'll be the focus of exam 1. I'm going to skip gene mapping until the next exam. So these are, in fact, the chapters. I do apologize a little bit. Everybody should have access to the current edition. These chapter numbers are the current edition version, what is it, the 7th edition of Pierce. So we should get up to here. Then we'll talk, I'm going to actually time swap linkage into where we begin talking about the biochemistry of the gene. And the reason for this is a linkage analysis is critical to understanding gene mapping. And so I think it's better to talk about the gene itself, gene sequencing, and then talking about linkage in the context of gene mapping, because this will help us a little bit later on when we talk about genomics, bioinformatics, and of course, any kind of forensic genetic analysis, which I think is important. So yeah, these chapters are right on the money. I should also point out, as of right now, it's too late. I've always threatened to do this over the years, and this year I actually did it. And if you poke your way into, if you actually read the syllabus, which I'm not telling anybody whether you are or not, you'll notice in the class format section, there is a little highlighted sentence that says, just to see if you're reading this, please send me your favorite song. It's worth 10 extra bonus points for the semester. And I'll put a playlist together. So for those of you who did read the syllabus and did respond with your favorite song, there'll be a new grading column in Blackboard, which you'll get your extra 10 points on. For extra credit points, and the playlist will be out Monday. So just to let you know, sometimes we do kind of crazy things with the syllabus, and so just you could check that out. And of course, my occasional comic, which I really like. This is, if you're in science and you don't know XKCD comics, this is kind of the classic XKCD comic book. So kind of keep you in touch with everything. You can come through here. I did add additional sets of links in the in basically the information up here. This is also in Blackboard, but I figured I would copy it here so that there was at least one additional spot to get this information. So so anybody have any questions up until now? Answers. Michelle always has a question. Come on.
Hector?
Yes.
So I did a blog post,
Excellent.
and I think I emailed you yesterday, and when I go to the page, I cannot see my post. Is that a normal, or do we have to put it like to make it see it? I don't know what's going on.
If you submitted, I saw your email this morning, and I will double check it. But if you submit it, there's a publish button, and if you hit publish, it should be posted on the main page. I will double check it as soon as we finish up the Zoom meeting.
Yeah, because I did publish it, but I don't know why it's not showing up.
I'll check it.
Okay.
Best I can.
And, okay. And another question. So where, like you just said, that the playlist is going off on Monday. So where we can find the playlist? Is that on the syllabus link, or?
I will put it on Piazza. It'll be it's a Spotify playlist, and it'll be, I'll post it on Piazza, just like I've been doing everything else.
Okay. Thank you.
You're welcome. So let's take a peek. A couple of years back, and I know some of you have looked at a few of these things already. I think this should work. Let's see, I have totally lost. Don't panic, it's only a cooking show. I have to reshare my screen. I really dislike terribly PowerPoint. But what I'd like to do is, there you are, okay. I always looked at these genetics problems, and one of the things that I like to reinforce when you're reading genetics problems, it is extremely important to, number one, read the problem and recognize how genetics problems are written. Secondarily, recognize that there are very often multiple ways to solve the problem. And last year, I discovered this really, really interesting question to me, and really enjoyed presenting it to students. So what I've got here is a notably non-genetic question, but one that utilizes math and the kinds of math that we use all the time in genetics. So I don't know if anybody's seen the movie The Martian. Basically, on one of the first missions to Mars, there's an accident and everybody's dead except Matt Damon, of course, because who would kill Matt Damon? And so Matt Damon survives, and one of the ways he survives is by taking potatoes and figuring out how to grow them in an environmental chamber on Mars, which allows him to survive. So this is basically, this question has nothing to do with Matt Damon and all about the math used to solve the problem. And the key element here is there are 100 pounds of potatoes, which are 99% water by weight. Here's where, obviously, what we're going to do is dehydrate them until they are 98% water. Okay, the scientific, that prepositional phrase for complex scientific purposes is totally meaningless. It just seemed like something to say. Why do you need to dehydrate them? Well, because it's part of the problem. So we're going from 99% water to 98% water. Once dehydrated, how much do Martian potatoes weigh now? So it's up to you guys right now. I want you to think about this for a moment. Hopefully you've got access to a pen and paper in front of you somewhere. Not going to ask you to turn this in, but I want you to take a couple of minutes and think about the problem. What do you know? 100 pounds of potatoes, 99% water. So how many pounds of water do you have? Not a trick question. Somebody yell out.
99 pounds of water.
99. See, it's not hard. This part's not tricky. This part's not tricky. Okay? We want to dehydrate them so that they are 98% water. So really, the question is, what is our total weight going to look like? Okay? So, anybody have an idea of how to solve it?
You set it up in a what's it called?
Got me.
What are they called?
You talking about cross multiplication?
Yeah, that yeah.
Mm.
I'm just messing with my screen here. Don't panic. So that's a possibility. Anybody have another idea? Oh, don't tell me you're all waiting on me to solve it.
Is there another way?
Oh, there are several ways.
Oh, all right.
Do you set it up as a proportion?
Ooh, that's an interesting idea. Try it by setting up a proportion. Of course, you do have to set up the correct proportion, right?
So, if we set up the proportion, isn't it gonna be like 99%? Is it like on a 99 over 100 or something?
Well, that 99 over 100 would be equal to the amount of water, right?
Mm-hmm.
Okay. I'm going to give you a clue, and there's a reason that I'm giving you a clue to an alternative method. What's a co-rec or a prereq for genetics? What course? Come on, you all took it.
Pre-calc?
Pre-calc. Hmm.
Or we can like do this. I think isn't it like we subtract 99 from 98, and then whatever we get that, and we put 98 into decimal form, and put the x value, and then solve it for x.
Ah, so what kind of a technique is that? What do we call that kind of an equation?
Linear equation.
And a linear equation or algebraic equation, right?
Uh-huh. Yeah.
Okay. So there's another method that one might use. Now, I think what I'd like to show you, and the purpose of this is not to confuse you. The purpose of this is not to put you guys on the spot, but the purpose is to get you thinking about the mathematical processes. Okay? So let's take a look at a couple of the different ways that we might want to solve this. And if I can figure out how to make the slides advance on Zoom, we'd be in good shape. There we go. So one, as a couple of people have suggested, is we can do some pretty much basic math. We know, for example, this is going to take me just a moment. I hate it. So we have 100 pounds of potatoes, which are made up of 99 pounds of water and 1 pound of potato, okay? But what we really want, I mean, you're not going to change the 1 pound of potato if we remove water, right? If we remove water, give me 1 sec. I want to get my pen here. Sorry about that. With my pen. There we go. Oh, I see. There's actually a command for this. Gotta love that. Okay, so the critical element is here. And in fact, I'm going to change the color of my pen here. Don't panic. Pointer options, pen color. Let's make it easier to see. There we go. How's that? Okay, so we've got, these are the two equations. There we go. We're a little red. So this really defines the problem a little bit better than kind of the way we were talking. Sometimes when we're dealing with percentages, and in genetics, remember, we're dealing with ratios. So just to draw the connection here, this is the reason for this particular problem set. And I can see, There we go. So the bottom line is we can do this by math, and we can see that this is really an unexpected result for most people, is that if we fill in what that question mark is, the only way to make it work is to have 50 pounds of potatoes, 49 pounds of water, and 1 pound of potatoes. So the answer, of course, is right here, is looking at 50 pounds of potatoes. All right? But this is just a rather simplistic way of solving the problem. The question, of course, is another way to do it. And some of you suggested that we could use ratios. And that's absolutely correct. So what we're really trying to solve is the question, and again, this is how you rephrase the question, is 2% of what number is 1 pound? And to translate this into an equation, here's where we use the ratio. So we know we want 2%, 2 of 100 times what equals 1 pound, and of course you get 50 pounds. And you can kind of test that after dehydration, we have 50 pounds of the potatoes, which again, from a verbal point of view, it's a little bit disconcerting because we use the term potato in 2 different ways. What we really talk about is we have 50 pounds of potato plus water, okay? Or what we can call potato matter and 49 pounds of water. So we really have to be very precise in the way sometimes we use words in different ways in different parts of the equation. Of course, we can also use an algebraic approach. And in the algebraic approach, we basically say, okay, we have 99% of 100 pounds is how much water there is before we begin the dehydration process. Ultimately, we're going to subtract 98%, which is our target, which is 100 minus some amount of water, right? So that's the amount of water after dehydration, okay? And both of these have to equal X, which is the amount of water lost from dehydration. So it's really setting up an algebraic solution, and I will leave it to you guys if you want to check my math, but the reality is each of these methods leaves us at solving the problem for finding X, leaves the problem at 50. So you really have 3 different ways to solve here. You can solve it using basic math and logic, is the way I describe the first solution. You can use pre-algebra by looking at the appropriate ratios, or you can use algebra, which is really where your pre-calc comes in, okay? And this approach is really, really important, especially when we consider some of the chi-square analyses that we've been talking about. So I want to give you a sense of the kinds of math and the kinds of problems, let me rephrase that, the kinds of problems that are used in genetics and the kinds of math that can be used to solve those problems. Now, this is the entire reason this is a Q2 course, okay? The Q2 course is here to emphasize quantitative solutions to biological problems, all right? I think you will find that I am more quantitative in my approach to genetics than many of the other, in fact, most of the other professors who teach genetics. And the reason is pretty simply that many of them are really spend a lot of their time doing molecular biology. And so they tend to be they tend to use molecular biology and focus on that more than the quantitative aspects of genetics. I think you need the quantitative to understand the applications of molecular biology. And to be honest, a co-rec here is organic chemistry. And you really haven't had enough biochemistry to really do your best in terms of understanding the molecular biology. So I prefer to focus a little bit more on the quantitative than some of the other professors. But we will talk about enough molecular biology so that you'll be dangerous and be able to understand some of the ideas of how we use a combination of quantitative technique plus molecular biology to do contemporary genetic analyses, because I think that's a really important part of what's going on right now. I think I showed this slide before. I absolutely love this slide. This is actually the original Punnett square. I scanned this from an original document when I was back at Penn State. This was actually a publication from the 1908 issue of Proceedings of the Royal Society of Medicine, written by Reginald Punnett. This was actually drawn by Punnett. I think he has lovely handwriting. And he actually put this together, and this describes the first of the Punnett squares. We generally don't think in terms of this as the original square. We think of the square as kind of this kind of a situation, right? This is the Punnett square everyone knows and of course loves dearly. And there's a reason that I'm drawing this for you. You've all done these a million times, probably starting in 7th grade biology. Takes me a little bit longer to do this on the computer. Although I think my handwriting's better like this than it is in real life. So compare these two Punnett squares. And the reason I like to show these is for the simple fact, we all know we have a 1:2:1 genotypic ratio, 1 homozygous Cap-A, 2 heterozygotes, and 1 homozygous small a. All right. Notice that I did not refer to Cap-A as dominant because we really don't have any phenotypic information here. Dominance and recessiveness refer to phenotype. Okay. I'm just showing 2 different alleles here. The reason that I like to show these side by side, if we were in a classroom, I'd be drawing on the whiteboard while this was showing as a slide in the on the whiteboard. I have been known on occasion to actually write on the slide screen. I get yelled at a lot for that, so that's a little scary. But the reason I like to show this is really to focus here on the heterozygotes, right? What's the difference between those 2 representations?
The orientation of the letters represented in the circles.
Absolutely. And what does that orientation tell you? What's the biological information indicated by that orientation?
Which parent had the little a and which parent had the big A and which child it went to.
Absolutely. And in particular, not just which parent, but which gamete came from the male parent versus the female parent. We don't see that here. We don't see this here at all. And it was the distillation of the differentiation of what we really call a reciprocal cross. It's reciprocal because here we've got a gamete from basically the one black parent, here from a white parent, and ultimately the white, and over here the black female. And we see a different source of the gametes. So a little bit later on, when we talk about epistasis, and we talk about epistasis from a reproductive point of view, and that is that in the mammalian species, the environment of the female body and the effects of the uterine environment on the developing embryo, and in particular, gene expression, is defined by comparing these reciprocal crosses. Because even though these two individuals are absolutely identical with regard to genetic complement, obviously in this case, if we have, here is an individual with the female gamete, when this is raised in a female environment, the genes from the paternal source are in the female environment. Here, genes from a maternal source are in the female environment. They behave differently. And this is the foundation of contemporary epigenetics. If we had not made this shorthand of defining our reciprocal crosses as the same genotype, we would have discovered the field of epigenetics 50 years earlier. So we have to be very careful when we're looking at any kind of experimental model, not to gloss over important information, or at least to assume, and again, I think some of this is cultural, because once upon a time, everybody assumed that there was no influence of the uterine environment on anything. Why? Because, as we will discover when we talk about some of the female scientists in the early days of genetics, their opinion was not valued just because of the cultural approach to women. So I think some of this was a result of just the cultural milieu at the time. But you can see how dangerous it is, that kind of an approach is, to understanding some of these scientific questions. One has to be really, really careful about either thinking things are unimportant or less important than. This is a mistake scientists make frequently in history, and one that unfortunately genetics has a really, really bad history of this. And we'll talk about this on occasion. And unfortunately, because of our digital nature, I will be posting about some of these things routinely as we move along through the course. So let's take a look in the video that you should have taken a peek at already. This is absolutely my all-time favorite question, and I do think this is one of the more important problems in genetics, and that is black coat hair. It's important for a lot of reasons. When we think about the names of phenotypes, very often we talk about coat hair. The terminology is pelage. Guinea pigs were a very, very popular choice for looking, as it was a very popular genetic model back in the 1940s through the 1950s. A lot of geneticists were breeding guinea pigs in their basement. In fact, Sewall Wright, who we will find a little bit later on, was the person who determined the mathematical analysis of linkage, without whom we would have no genomics information. There would be none of those very cute ideograms where we can assign genes onto particular chromosomes. Without the work of Sewall Wright, Wright was at the University of Chicago at the time. He had no lab space. He had a little small office in a very old building. And so he bred his guinea pigs in the basement of what was really a brownstone apartment in downtown Chicago. So just goes to show you how crazy geneticists are. And I say that as somebody who has about 30 vials of fruit flies in the garage because I don't want to take the chance of losing them if something goes wrong on campus. So what happens? Notice the way the problem is phrased. A pure line black guinea pig, the term pure line. Pure line implies, from the old breeding literature, pure line implies that black guinea pigs, no matter how many times they are mated and no matter how many progeny they produce, always produce black guinea pigs. The same thing can be said of the white guinea pigs, the pure line whites, okay? Now notice, we have a pure line black is crossed to a pure line white, and then the question says, what fraction of the black F2 is expected to be heterozygous? Notice that there is no mention at all, and I realize some of this was in the video, but I want to emphasize this again. It is extremely important, all right? No mention of the F1. The mention of the F1 really occurs here, okay? Pure line black to a white guinea pig, which means that you get an F1. In order to make an F2, it is assumed that you know you have to cross the F1s. So what percentage of the black are expected to be heterozygous? So anybody remember the answer?
2/3?
2/3, right? Why 2/3? Why not 50%?
Because it's out of only the black guinea pigs. We're not counting the the homozygous recessive white guinea pig.
Absolutely. Okay. I would say about, historically, about a third or more of introductory genetics students get that wrong and say 50%. So if when you looked at the original video or you think about the problem here and you immediately thought 50% because you're thinking 1 to 2 to 1, 2 heterozygotes, 2 out of 4 is 50%. Hopefully by now you realize that one of those 4 is homozygous small b, which means it's white, and the question asks what fraction of the black are heterozygous. So it's only 2 out of 3, and that's absolutely correct. But don't feel bad if you didn't get it at first, okay? This is a question of learning these differences. We're going to find out later on when we start talking about personal genomic analysis and genetic counseling, okay? This is really the kind of question that parents ask all the time. What is the probability of my child being affected if I am not affected? Okay, so in the vernacular of our pelage color example, if I have black pelage color, what are the chances of my child being homozygous recessive? Okay, a very important problem. And a lot of people get it wrong because they always assume 50%. They always assume the chances of you being heterozygous are 2 out of 4, not 2 out of 3. So very important distinction. So let's take an opportunity to take a look. Some of you may have done these already, but these are kind of the classic examples. I love this question here. If a black female guinea pig is test cross, it introduces this other idea of a test cross. This is an extremely important concept in genetics. Of course, if something is test crossed, what it means is an individual of unknown genotype is crossed to a homozygous recessive individual, a known homozygous recessive individual. So we have a black female guinea pig, will be test crossed. Obviously, if you take an unknown black female, it means what's its genotype? So this individual here, what's its genotype? You can do it.
Wait.
Yeah. Is it homozygous dominant, like for both big B's or something?
Well, we know because it's black, it must have at least what?
One of the.
So it must have cap B, right?
Yeah.
We don't know what the other one is, right? Because it could be homozygous cap B or it could be heterozygous. In both cases, it would be a black female guinea pig.
Yeah.
Right? In genetics, we use this underline to indicate either an unknown or we don't know the other allele. But we know it has to have a cap B because it's black.
Yeah.
It is test crossed to what?
A homozygous recessive.
Exactly. Who is phenotypically white.
Yeah.
Right? Easy peasy. So this individual produces 2 offspring in each of 3 litters. And one of these days, I'm going to correct this. I use this problem for a couple of different reasons. And in this particular case, it really doesn't matter how many litters there are because every single offspring is an independent fertilization event. So it really doesn't matter how many litters there are. It's a bit of a, what did Alfred Hitchcock call them? It's a bit of an albatross. It kind of distracts your attention a little bit from the problem at hand. So really, 2 offspring in each of 3 litters, meaning 6 offspring, right? Piece of cake. It's more indicative of my usual handwriting. Okay, so we've got 6 offspring. And the question is, what do you think the unknown black female guinea pig, what do you think her genotype is? All these offspring, all 6, are black.
Homozygous dominant.
Okay, so you think she's homozygous dominant, so she could have, and why do you think that?
All of them are black. They're kids.
Okay, but why does that matter?
Because if the female was heterozygous, then like half of them would be white.
Ah, right, everybody, these are not hard questions yet. Right? So the question is, this is typical genetics question. How confident are you that she is homozygous cap B? Well, some of you probably thought the same thing as Michelle, cap B, right? Shake your head, wave, pretend you're awake, right? But how confident are you? Very, a little, kinda. What do you think?
I feel like this is a trick question.
Why? Would I do that to you? Yeah, sorry. How confident are you? Take a minute, think about it.
Yeah, I can't think of any other answer, so
Ah, okay, excellent. So you're pretty confident. Most everybody's probably pretty confident. I'm pretty confident. I'm very confident that's the correct answer. Unfortunately, from a genetics point of view, if parents come in and ask, what do you think the probability is that my child will be affected? And you say, oh, I'm very confident. Bigly confident. Now, sorry, we won't get political. Do you think superlatives matter? At least in genetics, which is a Q2 course. The answer is, it doesn't matter what adjective I use. The question really is, when I ask, how confident are you? The question really is, what is the probability that I'm correct? So it kind of was a trick question, because the trick is, I'd like you to calculate the probability that you're correct. How would you go about that?
you set up a Punnett square.
We definitely need the information from Punnett Square. And somebody mentioned it a little bit earlier. If this individual was heterozygous, what are the chances that the offspring will be white or black?
50.
Right? 50-50.
Mm-hmm.
So we have 6 independent fertilization events. What is the probability that if the female was heterozygous, what is the probability of her having 6 black offspring? That's the question. So let's think of it in terms of flipping coins, right? I love coin flipping is like the best in genetics. Coin flipping is the best. Heads, tails, right? Flip a coin, 50-50 chance of heads or tails. Would you be surprised if you flipped a coin twice and you got 2 heads? It's not 50-50. What is it?
A quarter.
How'd you get a quarter?
1/2 times 1/2.
Right, because you have the probability of one coin flip and another coin flip, and anytime you hear the word and, you multiply probabilities. So if you flipped a coin 6 times, what would be the probability of getting 6 heads in a row?
I think like less than 2%. I
No,
got
I
like
don't want thinking. I want numbers. What would it be? Give me the
I
formula if you can't do it in your head.
I did like 0.5 like times, well, like 6 times, or I guess 0.5 to the 6, and
Ah, excellent. So a half to the sixth power, right?
mhm, I got like 1.56%, but I did it on a calculator. I didn't do it like that, so
Fair enough. Right? Half to the sixth power. So, and the percentage was what?
1.56%. But someone could check me on that.
I also got that.
Excellent. So 1.6%. So how, when I ask, how confident are you? How confident are you that she's homozygous? Are you 1.6% confident? Doesn't sound very confident to me. I certainly wouldn't bet my house at Atlantic City for on 1.6% chance. So how confident are you?
Would it be 98.4%?
Absolutely. I might bet the house on 98.4%. I would at least bet $1. Okay? So this is one of the really important problems, as simple as it appears. There are many different types of mathematical analyses that we will be using throughout the entire semester.
Wait, do you mean like we are 98.4% that the mom is heterozygous?
Know that she's home. In other words, this will occur. This result will occur 1.6% of the time this individual has 6 offspring.
Yeah.
Right?
Mhm.
So that means that it will occur. That means the probability that you are incorrect, that she is heterozygous, is actually 98.4%. And if she's not heterozygous, she must, by definition, be homozygous Cap B.
Oh, gotcha.
So again, you know, there's this combination of math, probability, and percentages and ratios that really are the crux of genetics. And that's why I say, if you can work your way through this problem, it's really only a matter of scale to be able to figure it out. With if you can do it for 1 gene, you can do it for 2 genes, right? The math is a little bit different. The ratios are a little bit different, right? And if you can do it with 2 genes, you could probably do it with 3. Take you a little bit longer. And if you can do it with 3 genes, you can do it with 20,000 genes in the human genome. Take you a lot longer. Unless, of course, you use some of the computational resources that are available to us. And in fact, if you get a chance to take bioinformatics later on as a junior or senior, both Dr. Luke and I teach bioinformatics. We take a little bit of a different perspective. Mostly, Dr. Luke does quite a bit of work with bacterial genomes because that is her area of expertise, and also some marine organisms, which is really great stuff. Me, I'm more a vertebrate guy, so I tend to deal with eukaryotes. As I've said before, I fundamentally don't believe in prokaryotes. They were too dumb to get together and make a eukaryote. So, I tend to be more, I focus more on vertebrates, including human genomics and human bioinformatics, which is a little bit different. So we alternate years. We have a lot of fun together, and we use some of the same exercises back and forth between us. But this is really the crux of the math. This is it. So, sorry. So let's take a peek at a couple of other problems. This problem set, I am going to, let me figure out how I'm going to do this. There we go. Aha, good. I am going to share, oops, sorry, I just missed some chat stuff. Oh, good. Sorry, I missed some of the chat items here. When I have full screen with only one monitor, I can't see the chat stuff. Good. With regard to the lab, let me just answer very quickly. With regard to getting the pictures of the corn, I just received late last night some stuff from Lab Archives, and they have a suggestion on how to fix it. Nobody seems to know exactly why it won't work, but I'm gonna take their suggestion, check it out, and so sometime this afternoon, just revisit it. Don't panic. Nothing's gonna be late. Everybody take cleansing breath. See, very nice. Do the tree pose for about 5 minutes. You'll be fine. All right. Everybody know what the tree pose is? Who does yoga? Somebody. On what? How many feet? 1 foot. Yes, I got a few. Okay. So go stand on 1 foot for about 5 minutes. It's good for you. So let's see. Let me quick share my other screen. Don't panic, I'll find it. Hark. Oh, I know. Okay, so first things first. Let's try this one. So a quick note, if you go to the resources tab, you will see the first problem set with all the problems we've been talking about right here. And so what I'd like you to do is to attack each of the problems that are in this. There are a handful of problems up until 2 or more genes. You can fill these out in a Word document, whatever makes you, whatever makes things, oh, I hate Macs. The wheel works in opposite directions, sorry.
So, sorry.
Yeah, sorry about that. These computers are a pain in my butt. So basically, answer the questions. There's an example of lethal alleles and, of course, multiple alleles with rabbit coat colors and a final guinea pig. So up until this page 4, get those done. There will be this afternoon a Dropbox and Blackboard for you to submit your answers. Some people, if you want to just do them by hand and take a picture and upload it, that's fine with me. So pretty simple, but these are classic genetics problems for you to take a shot at.
If
Now,
we have them, do we have to copy down like the question and stuff, or like everything from the slide? That way you know which one we're talking about. Or should we just do like the label of the slide and then our work?
whatever you want, I can figure it out.
Okay, so it's just those 3 or 4-ish problems, basically, right?
Exactly.
Okay. Just
Yep, up until page 4.
And is there like a due date on the problem set for this?
We'll let the due date will be. Some of you have already done them, I know. So the due date, let's just say Monday. Let's get them done over the weekend. So that way we can move forward with pedigree analysis next week.
Okay, cool.
I do want to spend just a brief moment talking a little bit about chi-square tests. Chi-square is something that is extremely important in genetics. I know that Lab instructors have their kind of own view of doing this, and I also have kind of my own little take on it. Most of you have seen or at least probably looked at the video by now. But what I also have, which I'm going to stop my share and go to another share screen here. If you were Microsoft Excel, I have a spreadsheet that I put together, and I will put the spreadsheet in the resources tab of Piazza. I shared this spreadsheet with people who are in my lab section, but I want to give the opportunity for people who are not in my lab section to go ahead and have the spreadsheet as well. The spreadsheet is, oh gosh, I'm just looking. I don't have any way to annotate on an Excel spreadsheet. Love that. What a pain in the butt. Swear. Oh, maybe, wait, don't panic. I'm going to try something. Oh, I do. I'm so excited. All right. Oh, am I excited? Wow. I didn't know it would allow me to do this, so just hang on for just a moment. So what I guess what I've done here is try to help you out by setting up a spreadsheet to actually do the chi-square analysis for you. You can see we've got a monohybrid and a dihybrid cross. We have the expected ratio. By now, this should be getting automatic with you. If you do an F2 monohybrid, F2 single gene cross, we have expected phenotypic ratios of 3:1. 3 will be the dominant phenotype, 1 will be the recessive. In a dihybrid cross, the expected ratio, again, 9:3 to 3:1. 9 is the double dominant, dominant recessive, recessive dominant, and only 1/16 of the individuals will be of the recessive phenotype. I have a column here for your observed ratios. In this case, this is a made-up example for the F2 monohybrid, 306 dominant phenotypes, 94 recessive, of which the total is 400. These data for the F2 dihybrid cross are actually from Mendel's experiments, so give you an idea of what Mendel actually did. I always calculate an observed ratio. This is not something that is noted in the textbook. It is not something that is noted in the lab manual. But I will tell you that everyone I know in the real world who does genetics automatically, before doing a chi-square, automatically calculates an observed ratio. How do you calculate the observed ratio? Well, the answer is you take the smallest numerical category, phenotypic category, divide it by itself, which gives you 1, and then divide all the other numbers. So this would be 306 by 94. And of course, I round to the nearest integer. So we can see that even though 306 and 94 is not exactly 3:1, this would be 300 to 100, obviously, would be our expected. It still ends up being 3:1 if you create an observed ratio. If we look down here at the F2 dihybrid, these are Mendel's data, 315, 108, 101, 32, and you can see his actual phenotypes here. Note the observed ratio, again, rounding to the nearest integer, 10 to 3 to 3 to 1. All right? So the question that is always answered by a chi-square is, do deviations between our observed ratio and our expected due deviations occur because of some physiological, genetic, biological reason, or is it due to random chance? That's the critical element. And of course, the answer right here is you can tell this by subtracting your observed minus expected individuals. Here it's 300 minus 306 is 6. Squared is 36. And of course, we divide by the total number of expected individuals, 0.12. Same thing here. We find the chi-square number of 0.48 rather than making you go to a table or a graph of chi-square values. There is a way to have Excel actually give you the p-value, okay? And in fact, if you, oh, you're not gonna let me do it, are you? If you click on the red triangle in the corner, it will give you a more detailed explanation of this calculation. So the bottom line is the deviation of 306 to 300 is actually the p-value, meaning the probability that this is a real, this is no different from the expected, is 0.488. That is, and this is, by the way, in a percent, so we have a 90, somebody do the math, 99.5% confident that these individuals are not different due to some functional issue. If we look down here and we look at the observed for Mendel's work in the F2, he observed 10 to 3 to 3 to 1 as opposed to the expected 9 to 3 to 3 to 1. And so what is the real difference here? And notice that in Mendel's work, Mendel comes out with a p-value of 0.92, which means we are basically 99% confident that these, the 10 to 3 to 3 to 1, is only different from 9 to 3 to 3 to 1 by random chance and not due to any functional difference. So Mendel's work, you could see, was incredibly accurate. The one thing that students always get confused about, and rightfully so, is the concept of degrees of freedom. And you will find a series of notes in the other spreadsheet tab about the idea of degrees of freedom. And it's really important, I think, for you to understand at this point that the concept of degrees of freedom is more complicated than just subtracting 1. It happens to be true in our case that we have 2 phenotypic categories for the F2 monohybrid cross, which means 2 phenotypic categories minus 1 is 1 degree of freedom. Here we've got 4 phenotypic categories. 4 minus 1 is 3. It is absolutely true that for our purposes, you can only subtract 1. But do not begin to think that in all cases, degrees of freedom are simply calculated by subtracting 1. They are not. They are not.
Barbato,
Yeah.
I have like class in like 10 minutes and I have a couple questions.
Yes.
So from our first
Well, wait a minute. Let me finish, and then we'll be able to move forward. Okay. Okay. Yeah, I see we're like at the ending point. So anyway, I'm going to make these available, make this spreadsheet available to you guys. The people who are already in my lab have it. Everybody else will have access, and feel free to go ahead and use it. Okay. So Michelle, you had a question.
So from the earlier with the potatoes, how do you know that the 1 pound of potatoes is constant throughout? Or
Because
like,
we're only dehydrating, which by definition is removing only water.
so like you don't like, so like the weight of the potatoes itself wouldn't shift or anything like that. It's still just 1 pound. because you're only messing with the water in it.
Right. So you've got water and what I called in there potato matter. Okay. So if you make a distinction between the word potato being potato matter plus water or dry matter plus water, right? Because if you remove water, the rest is dry matter. So if you make a distinction between dry matter and water, and the sum of dry matter plus water equals potatoes, okay? Again, it's all about constructing the mathematical relationship.
Gotcha. And then the problem that you did in the video with the N over K in the parentheses with the exclamation point.
Yep.
So the question when it was like 3 whites and 2 black guinea pigs, does it matter which number we put on the bottom for K?
No, it just matters in the context if you're doing black or white.
Okay, I
So
just
one
didn't know.
for black and one for, you'll put, I forget what the numbers are, but it'll be like 3 for black, 2 for white.
Yeah, so like if I was just trying to find the outcome for 3 whites and 2 blacks, then it doesn't matter if I put 3 or 2 in.
Correct. You just have to remember which one because there'll be 2 different probabilities. Yes.
Okay, cool. All right, thank you so much. We know.
Yeah, no problem at all. Good luck in your next class.
Okay, got you. Thank you.
So as usual, I'm going over time. So anybody, if you got to go, if you got to hit the road, please feel free. If you got a question, I got a little bit of time here.
Professor, I
Yes.
just want to please check the blog post and let me know if it's showing up so I can make it happen.
As soon as we get off this Zoom call,
Okay.
I'll check it.
Okay, thank you.
All right, everybody good?
Have a good
Yeah.
day.
Have a great weekend. Go apple pick.
And you do gonna post the playlist on Monday, right?
Yes, I am, as a matter of
Okay.
fact. All right, you
Thank
guys
you. Have a
take,
good day.
you too. Take care, everybody. All right, have a good one, and we'll see you next week.